Hello, welcome to this session about research and treatment development for unknown ataxia family history. It is my pleasure to introduce our speaker today, Dr. Martine Tétreault, who aims to contribute multiple omics to the study of genetic etiology of neuromuscular and neurodegenerative diseases. Dr. Tétreault's career has been highly productive with over 70 publications. She has received funding from several Canadian and American organizations, as well as a grant from the Canadian Foundation for Innovation to establish a translational platform to study rare and common neurological diseases. She also received the David Green Award for Emerging Researchers, the Early Career Award from the CHUM Research Center, and the Fonds de Recherche du Québec Junior 2 Salary Award. And with that, Dr. Tetreault, thank you so much for joining us today. The floor is yours.
Thank you. Thank you so much for the invitation to talk about research advancement and treatment development for unknown ataxia with family history. So I have no conflict of interest to disclose. So first, I will just do a brief introduction on some ataxias and some concept that will help with the rest of the presentation. So ataxia is basically a symptom which corresponds to impaired of balance and coordination. Many aspects can be impacted by ataxia, for example, walking, balance, hand coordination, speech that we'll call dysarthria, swallowing, so dysphagia, or eye movement that we call nystagmus. Those symptoms result from damage to the cerebellum or its connection. Cerebellum is a part of the brain that is really important to regulate timing and thought of action. It can integrate vision, sensation, proprioception, and other environmental cues that are required for having smooth movements. There's different causes of ataxias. Some are categorized as acquired causes, which can develop symptoms following infections, autoimmune disease, other exposition to toxic agent, etc., can lead to the development of ataxias. On the other side, we have genetic causes where there's a change in the DNA code that will lead to development of disease. There's different mode of inheritance. Can be dominant, recessive, X-linked, or even a genetic variant in the mitochondrial genome. A lot of the cases with familial history will be in this category of having genetic causes. There's more than 100 forms of ataxia that has been described to date. This number can be even higher when you consider ataxia as a secondary symptom. And for hereditary ataxias, there's around 10% out of 100,000 that are affected by these diseases. And then we have the idiopathic causes. So in this category, it's patients that are presenting with ataxia symptoms, but we actually do not know the cause of the symptoms. Usually there's no clear family history. And with further investigation, those patients we can find that those patients might have an acquired causes or even genetic causes. Currently, in the clinical diagnosis setting, a patient will come to the clinic, will present signs of ataxia. The first thing that the clinician will try to determine if there is any familial history and a possible mode of inheritance. If there's familial history of ataxias, then usually we will go further on the genetic testing, and it's the mode of inheritance that will guide the test and the genes that will be studied. If there's no familial history, then it will be considered a sporadic ataxia. And then with some investigation, we will determine if there's a possible genetic cause and move on to genetic testing, or if there's no genetic cause suspected, try to find some acquired event that could have led to the development of ataxia. So when to look for a genetic cause? So one of the main factor is a positive family history. This is usually a green flag thing that there's a genetic cause involved. Other factors include their similar clinical presentation to other genetic syndromes, a chronic or progressive disease. If there's consanguinity in the family, we will usually suspect a genetic cause, or if there is an increased number of cases in founder population. For example, here in Quebec, the French-Canadian population, we see an increase of certain type of ataxia, the frequency of certain type of ataxia, or an increased frequency in a specific population that is not necessarily known for founder effect, then we will suspect a genetic cause. So patient will go for a clinical evaluation. This evaluation includes a physical or neurological exam. Sometimes we'll have imaging, such as MRI, to see if there's any abnormalities in the brain, in the cerebellum of the brain, and can also involve some biochemical analysis to look at lipids or other metabolic factors that could have influenced the development of ataxia. Then, if we suspect a genetic cause, we will do genetic testing, usually specifically looking at ataxia genes. And despite the clinical evaluation and the genetic testing, there's still around 30% of ataxia patients that do not get to have a clear diagnosis with genetic finding. Those patients are often included then in a research project where we can further investigate the genetic cause using other approaches that are not yet available in a clinical setting. However, it's important to note that for ataxia patient with genetic cause, the family history is not always clear. This can be due to late onset disease, for example, where we are not able to gather enough information about other family members or parents to determine if there's a familial history of the disease. We also observe incomplete penetrance. Some mutation are not acting, are not giving their 100% all the time. So in the same family, we can see different severity or different presentation and canvas an ataxia that caused by a repeat expansion in the gene RFC1. We'll talk about it later. It's one example of that, where we have family member that have a severe ataxia with sometimes a sensation problem, and other family members has basically only a cough. And this brings me to the third point, where we have variability in clinical presentation. So siblings sharing the same genetic cause can have a different age of onset of the disease or level of severity. So this makes the study of ataxia a lot more complicated. This brings me to talk about those challenges when we study ataxias. So like I said, there's some heterogeneity, some variability. There's clinical variability. So what we mean by that is different in severity, age of onset. And there's also a genetic heterogeneity. So we can have several genes that will lead to the exact same clinical presentation. So for a neurologist or a clinician seeing ataxia patient in their clinic, it can be very hard to determine which genetic cause can be involved. Ataxia genes are also very large and highly polymorphic. So they can have a lot of genetic variation that are benign and that does not cause disease. So this also makes it more difficult to establish a clear genetic cause for ataxia patient with family history. If we take an example, the gene SIGN1, which it has 146 exons. An average gene has about 10 exons. So since we have a lot more, the gene is a lot bigger, it tends to accumulate more variants and makes the interpretation a lot more difficult. Another example is the gene SAXIN that causes the Charlevoix-Saguenay ataxia. It's a 10 exon gene, so about the average number of exons, but one of the exons is 12.8 kilobases. An average length of an exon is 147 base pair. So it's more than 80 times bigger than the average exon. Again, because of this large size, we accumulate more genetic variation. Most of them are benign, but it just complicates the interpretation of genetic data for familial history, ataxia patient with familial history. Another challenge is there's a lot of genetic changes that are observed in ataxia. So we can have just a change in the code that will change one letter in the code and lead to the development of a disease. And we can have some variation that are a lot more complex. If we think of repeat expansions, for example, that are involved with, that are causing many ataxias. So this again requires multiple tests, and it can take longer before we identify a genetic cause. And finally, one important challenge that applies to ataxias, but also other rare diseases, is that despite a lot of research, basic scientific biological research, we still don't understand the genome, its function at a level enough to interpret all the genetic variant we can find. There's also some technical limitations that makes us unable to find certain type of variants. So to date, our capacity to identify variants is very high, but our ability to interpret and give a clear results of those variants is still limited. So I will start by talking about the genetic analysis of unknown ataxia with family history and the most recent development in sequencing and how this can help to improve our ability to diagnose patients with ataxias. So genetic studies have been forever driven by technological process. It's very progress in different technology that enables us to identify more causes. 20 years ago, 20, 30 years ago, we were using what we call linkage analysis to find which region in the human genome was responsible for the disease. But this was only possible when we had large families with multiple generation. So as you know now, it's very rare to have families with 5, 6, 8 children. Usually we have 1, 2, 3 are considered big families. So we don't have statistical power to use this approach anymore. But since the early 2000s, we have a pretty good draft of the human genome, and this has led to development of new technologies that we call next generation sequencing, and more recently, third generation sequencing. And this enables the detection of more variants and does not require to have those big family anymore. So here, if we talk about next generation sequencing, and for the following slide where I'll try to explain those sequencing technology, I would like you to think about the genome as a puzzle. So each puzzle piece corresponds to a piece of DNA that we have sequenced. And when we analyze the sequencing data, we have to put back all those pieces together to get the full picture of the puzzle, so the full picture of the DNA we have sequenced. So for exome sequencing, we are looking only at DNA regions that contains the information to produce proteins. It's about 1 to 2% of the entire genome. Like if we look here at the puzzle, we will be looking only at the part of the puzzle where we have a clear image, we have the clear information of what this puzzle is showing. So here, a DNA molecule. For whole genome sequencing, we're looking at the entire DNA, so the exons that are producing the proteins and the introns. So if we go back to the puzzle, we will be looking at the entire puzzle, even the white space, which is the white space where we don't really get an information on what's the image in the puzzle. The pieces look pretty much the same, where we don't really know what they are. They don't give us clear information. We would have to look at it more closely to see if there's differences between those pieces. So this is what next generation sequencing is. We're looking at the DNA, either a very specific region of the DNA or at all the entire genome. For both exome and genome sequencing, we will obtain sequences. So again, puzzle pieces, and we will put them together, and then we will compare them to the image on the box of the puzzle to see if there is any difference between our puzzle and the image that we're supposed to get. So here, we're in terms of DNA, we're looking, is there any differences in the DNA code, and we identify those. And once we have those, we try to interpret those differences in the context of the disease, here ataxias. So is this genetic variation can lead to the development of ataxia? In which gene is it present? What is the function of this gene? Could that explain the clinical presentation we're observing in the patient? For each patient, we're looking at individual causes. So it's a little bit like trying to find a needle in a haystack. So it can be very quick, but it can also be longer if the genetic variation is more complex. Here, I want to give you an example where exome sequencing led to finding a genetic cause. So we were studying a family, two brothers affected with the juvenile onset of recessive ataxia. The two brothers had a different phenotype. The younger brother had a more severe presentation and an earlier onset. He's the one here you can see in the pedigree with the red arrow. So we performed exome sequencing only in this individual. So again, this is an advantage of those next generation sequencing approach where we don't need data on the entire family. And using exome sequencing on this patient, we identified a variant in the gene PMPCA. At the time, there was only one scientific publication linking the gene PMPCA to an ataxia phenotype. So because of the similarity with the case in the literature and the genetic finding, we were convinced that this was the cause of the disease for this family, and we were able to establish a diagnosis for the two affected brothers. So for next generation sequencing, both exome and whole genome sequencing have successfully identified genetic causes for patients affected with ataxia. Those technologies have led to an improving of diagnosis and leading to 30 to 50% of cases with a molecular diagnosis. We do not require having data on entire families. We can do single cases and still identify genetic variation. But despite the success of this technology, there's still a large proportion of patients with familial history that remain without a diagnosis. One important reason for that is that we identify variants that will be classified as what we call variant of uncertain significance, or VUS. You're going to hear that a lot during my talk. These variants are that we know there's a genetic difference. We know there's a difference in the code, but we are not able to say if this variation in the DNA code it is benign or lead to development of ataxia symptoms. So we're not able to diagnose the patients. We need further investigation to confirm the impact of this variant. Another reason is that we can look at the entire genome, so the pieces of the puzzle where we don't have a clear image, but those correspond to intronic variants, but we're not able to interpret those variants clearly and know what they're doing. So again, those variants, we're able to detect them, but we're not able to assess if they will cause the disease or no. And then there's other variants, such as repeat expansion, which are very important in the context of ataxias, that are difficult to identify using next generation sequencing. And I will explain in the next slide why and what is the solution to this approach. With more recent, so what we call third generation sequencing or long read sequencing approaches. So here I will come back with the analogy of the puzzle. We want to compare the next generation sequencing that I just talked about, which use a short read. So we sequence small fragments of the DNA at a time, versus a long read sequencing where we sequence large fragment of DNA. So think about the puzzle again. So the short read is like if short read sequencing is like if we have a puzzle of hundreds or thousands of small pieces and we need to put them all together. Since the pieces are small, there's a risk that two pieces are very similar and we don't really know where they go in the puzzle. We don't know where to place them because they look exactly the same. So it's the reconstruction of our puzzle is more difficult and less precise. Long read sequencing is like a puzzle for children where we only have a few pieces. The pieces are big. They have a lot of details. So it's easier for us to put them together, to reconstruct our puzzle, and to visualize the image that we're trying to form. So this is basically the difference between the short read sequencing and the long read sequencing. So using long read, we are able to have less pieces of the genome where we don't know where to put, less gap, we have more information, and this gives us the opportunity to detect variants that are not detected using the short read sequencing. It's a pretty recent technology, long read sequencing. In 2023, it was identified as the method of the year by Nature Methods. And since then, it really has taken a huge place in genetic studies of different diseases, including ataxias, and how it can help to the diagnosis at many levels. So as I mentioned before, we think there's at least 30% of patients affected with ataxias that are without a molecular diagnosis. So how can long read sequencing help? So because we have those big puzzle pieces where the assembly, so the reconstruction of the genome for each individual is a lot easier. Because of that, we're able to detect more complex genetic variation, such as structural variants, including repeat expansions. In the context of ataxia, this is really, really important. Another aspect that will help in the diagnosis is that we're able to phase variants. So in the context of recessive disease, the patient will receive a genetic variant from his mother, a genetic variant from his father. So we have two variants. We're able to really assess if those variants are causing the disease. We need to determine if if they were transmitted by both parents or by a single parent. So this is what we say about phasing. Typically, especially for neurodevelopmental diseases or congenital ataxias, we will sequence a trio. So having data on both parents will enable us to determine if which variant was transmitted by the mother and which variant was transmitted by the father, or if both variants here were transmitted by the same parents. But when we're in presence of a later onset disease, we usually don't have access to parents to get DNA. So we're not able to do this analysis and determine the segregation of the variants. That's where long read sequencing can help us, because those long read will help us determine by sequencing only the affected patient if the variants were transmitted by both parents or by a single parent. I will present an example of that to try to make it clearer. So here we had a male patient, was diagnosed with progressive ataxia in his late 50s as episode of imbalance, ataxic gait, nystagmus. There was atrophy of the cerebellum at the imaging. With the clinical presentation, we were suspecting a mitochondrial disorder. We did a whole genome sequencing, and we found two variants in the gene SPG7. SPG7 is associated with autosomal recessive spastic paraplegia, as well as recessive cerebellar ataxia. So it seemed to be at the symptom level, it seemed to be a good fit with our patient's presentation. So we had one variant was introducing a stop codon, and another was just changing the amino acid. So we had two variants in SPG7, but we did not have access to the parents to confirm that both variants were inherited separately. So we did long read sequencing. So here at the top, you can see an image of the gene, and we have produced amplicons to amplify just this gene. We just wanted to look at SPG7, and we sequenced SPG7 using long read sequencing, and we were able to determine that the missense variant and the stop gain variants were inherited by both parents. So the mother transmitted one of the variant and the father transmitted the other variants. So with this information, we were able to confirm SPG7 as the cause of the ataxia for this patient. And the patient finally received a genetic diagnosis for his ataxia. Long read sequencing is also able to detect repeat expansions. So there's a lot of study that showed that it was successful to detect that. Those expansions can be in different genes, although challenging to detect using previous method. Long-read sequencing has proven to be efficient to detect them and determine the size of the expansion. More recently, in 2023, there was a repeat expansion that was identifying the gene FGF14. I worked on this project with Dr. Bernard Bray here in Montreal, and this is a family. Those families of late onset ataxia from French Canadian descent were under investigation for more than a decade before we actually found this expansion in FGF14. So we were able, with 3 family, we sequenced the individual that you can see here in the red box, and we actually found, following whole genome sequencing, we found some weird puzzle pieces that we were not able to put back together, and we're suggesting that something was happening in this region of FGF14 where we had a repeat expansion. So we use long read sequencing to try to look at it more closely, and we actually found here in the patient where we had those longer repeat counts, 400 around 400 repeats, compared to control where we did not see that at all. So long read sequencing confirmed the presence of a repeat expansion in the gene FGF14, and we were able to have a better estimate of the size of the expansion in those patients. So after 1 or 2 decades of investigation, those families finally had a genetic diagnosis, and it's turned out that repeat expansion in FGF14 is now one of the most common form of ataxia here in Quebec. So long-read sequencing can clearly benefit for the diagnosis of ataxias. We get a more accurate picture of the genome, so big puzzle pieces versus small puzzle pieces. We can identify structural variants and repeat expansion that we're not currently able to detect with accuracy using other methods. And we're able to phase variants, so determine the inheritance of those variants in terms of maternal allele or paternal allele. So long-read sequencing has the potential to improve the diagnosis for patients affected with ataxia without a known cause. For now, it's mostly used as a research tool for family that didn't have a cause, but hopefully there's a lot of projects in the US and Canada and elsewhere in the world to do proof of concept and to try to move this long-read sequencing approach into clinical setting. However, there's still important obstacle that remains despite this sequencing technology development. So we still have variant of uncertain significance where we're not able to determine the impact. And we also have a lack of information on how some of the genetic variation can result in ataxia. So we need to find more other research investigation to be able to classify those variants as disease causing. So for that, we can be looking beyond DNA. For example, RNA. So what is RNA? So basically, RNA is a copy of the DNA, but without the introns. So we have the DNA where we have a puzzle with all the pieces. When we look at the RNA, we will concentrate only on the pieces where we have an image, so that are important to produce a protein. And what is important to know compared to DNA, so DNA is present in all the cells. It will be the same in all the cells in terms of genetic variation, but RNA can be turned on or off depending on the cell types or the tissue we will be looking at. So if we sequence RNA from blood, we will see genes that are turned on in blood that might be different from the genes that are turned on in the brain. However, it's very difficult to get access to brain samples. We won't do a brain biopsy just to do RNA sequencing. So usually we will use other tissues. The sequencing itself is very similar to exome sequencing. So it's pretty much the same technique. And how can how can RNA-seq using long read or short read sequencing can help? So we can look at genetic variant that would impact splicing. So splicing is the process where the exons, so here in blue, are removed and where we keep only the exons, so the green region of the DNA. And we will look at genes that are important for the disease by seeing which one are turned off or turned on. As an example here, again, was a patient affected with a late onset ataxia. The onset was around 73 years of age. The patient presented with dysarthria, progression leading to ataxic gait, some episodes of nystagmus. Again, MRI showed some atrophy of the cerebellum. We did a whole genome sequencing, and we found a variant in the gene L4. L11.4 is associated with autosomal dominant spinocerebellar ataxia, so SCA34. So again, the phenotype, the clinical features described in the literature and what we were observing in our patient were very similar. So it was an excellent candidate for the ataxia in this patient. However, the variant we found was at a splice junction. So we were not able to determine with the DNA alone if the variant was causing the disease. So we performed RNA sequencing, and what we actually saw in the RNA is compared to the normal RNA here in blue, what we observe in the patient is that the exon number 4 was missing. So our difference in the DNA code at the DNA was actually resulting in the loss of one complete exon at the level of the RNA. So with that, we were able to confirm that this variant was impacting the function of the gene and that L4, so SCA34, was the diagnosis for this patient. There's also promising studies looking at methylation. So what is methylation? It's a chemical process where a molecule called a methyl is added to the DNA. So methylation can impact expression by turning them on and off. So it has a very important function. There's a lot of studies that shows that it has an important role in ataxias. Friedreich ataxia, for example, SCA3, SCA2 have shown some abnormalities at the methylation level. Methylation can be involved in the mechanism leading to the development of ataxia, but can also be associated with variable severity among patients with the same genetic variation. There's also a growing interest as using methylation as a therapeutic target. If we talk about Friedreich ataxia, so FRDA, Friedreich ataxia, is a repeat expansion disease. The fact that we have this expansion in an intron of the gene leads to a hypermethylation. So we have an increased methylation at the DNA level, close to the expansion. And this results in the gene frataxin to be turned off. So we have a reduced expression of the frataxin. We know that the level of this methylation is associated with a severity. So for Friedreich ataxia, the size of the expansion, so the bigger the expansion, the more severe, but also the level of methylation seems to also be associated with severity. And there are currently some studies that are looking at using treatment that will target this methylation, so reverse the increase in methylation to try to increase to try to turn on the gene frataxin and reduce or cure the symptoms associated with Friedreich ataxia. So this is still it's still at the research stage, but it's an interesting therapy strategies that might become available in the future. So future direction in terms of sequencing. So a lot of the labs, including my research labs, we're trying to use a multi-omic approaches. So we combine different sequencing by looking at DNA, looking at RNA, looking at methylation, combining long read and short read sequencing to try to identify the genetic causes in those ataxia patient with family history, where we are pretty sure that have a genetic component, but we have not been able to identify yet. So this is where the field in terms of sequencing is going, is more toward a multi-omics approach. Again, I will talk a little bit about model systems. So what are model systems? So they include cellular models, so iPSCs, so cells that we have reprogrammed, organoids when we're trying to reproduce an organ, gene editing models. There are simple animal models like yeast, worms, flies, or more complex animal models like zebrafish or mice, and even human-based analysis. How can this model system, what are the applications of those model systems? So it's important for personalized medicine. So I talk a lot about variant of uncertain significance. So using those models, we can look at the impact of the variant in different contexts. We can also use those models for looking at responses to therapies. We can learn more about the mechanism leading to disease, identify biomarkers, again, that will be helpful to look at responses to therapies or to cluster patients that will be more, cluster patients that would be more relevant to different clinical trials, but can also use those models to develop new therapies and test drugs. I will give you here an example of personalized medicine. So we had a patient affected with a late onset progressive ataxia. He was diagnosed with a genetic cerebellar ataxia, onset around the age of 45 years, at ataxic gait, dysarthria, dysphagia, hyperreflexia. We did sequencing and uncover a genetic variant in the gene ETPR1, so it's a calcium channel gene. And this variant was just changing one letter in our DNA code. We were not able to assess based on DNA alone if this variant was causing the disease. So it was classified as a variant of uncertain significance, and no diagnosis was made for this patient. So we decided to use a gene editing approach. So we use a cell line here that has nothing to do with ataxia. It's a kidney cell line, but has where the gene ETPR1 is turned on. Using gene editing approach, we incorporated the patient's genetic variant into the cell lines, and ETPR1 being a calcium channel, we measured calcium. Using this model, we can see in blue, which correspond to the patient's genetic variation, we can see there was a decrease of calcium in the cell lines. So we were able to assess that this genetic variation impact the calcium release in the cells, and thus the cell cannot function normally. And with this information, we were able to establish a diagnosis for this patient, and confirming that ETPR1 was actually the cause of the ataxia for him. Those models are also important to understand disease mechanism. Here is an example of a model for RFC1, so CANVAS. So the gene CANVAS was identified in 2019, so it's quite recent. Despite that, it's still not clear how this expansion leads to the development of the disease. So we know that it's a very complex expansion. So it's a pentanucleotide expansion, but the pattern of the expansion is not only an expansion, but the pattern of this genomic region also change in a disease context. So it's a very complex disease, and this is partially why the phenotype, the mechanism is not well understood. One of the first symptoms before having any ataxia symptom is coughing. A lot of patients report having a cough before being diagnosed with CANVAS or having any ataxia or imbalance or neuropathic symptom. So we use, in collaboration with colleague Eric Samaru, we use a zebrafish model where we turned off the gene RFC1. So we were able to show in this model that the zebrafish had early cerebellar defect at an early stage, And we also saw using single-cell sequencing of the brain of those fish, we were able to show that we had a loss of neuronal progenitor cells. And this is very important because all what we were observing are actually at the developmental stage of neurons of the brain. So this model support an important role of RFC1 in neuronal development, which was not what we expected when we think about RFC1 canvas being a late onset disease. However, discussing with clinicians, we actually learned that a lot of patients were complaining of coughing or other symptoms many years before the actual diagnosis or of ataxia. So there's a possibility that actually this neurodegeneration or the impact of RFC1 expansion starts earlier in life, but the development of the disease itself happens later at a later onset. So this opens a new avenue of how those late onset ataxia gene might have an important role in the development of the brain and the neuron system itself. So more investigation are obviously required to clarify this aspect, but it's something that other groups are also observing for other late onset ataxia genes. For the final part of the talk, I will try to do an overview of treatment development and research that are done in this aspect for ataxias. So for ataxia of unknown cause, treatment usually will focus on managing symptoms and improving the quality of life. So this can be like physical therapy, occupational and speech therapy, medication that will manage muscle stiffness, pain, or depression, or adaptive devices like canes, walkers, or wheelchairs that will be more to managing those symptoms and not really cure the ataxia. Most of the ongoing research for treatments required some sort of genetic diagnosis to be to have some targets to be able to work on those specific targets in order to try to cure the ataxia phenotype. And those include drug repurposing. So where we're using drugs that are already approved by FDA, for example, and we're testing them in different models, the model system I presented earlier, to see if we're able to rescue some of the ataxic or neurodegeneration phenotype. Gene editing, RNA therapies, such as RNA interference, antisense oligonucleotides, stem cells, and other pharmacological therapy are all avenues that are being studied currently in research for the treatment of ataxias. For gene editing, for example, the principle of gene editing is targeting mutant alleles to enable precise and permanent gene correction. So we're trying to correct the error in the cells so that the cell can function normally. So the main approach used is CRISPR-Cas9. Recently, this approach was applied to SCA3 using iPSC. After correction, cells develop into normal neuronal cells and maintain electrophysiological properties. The same approach was also applied to SCA1 fibroblast and showed decreased expression of disease-related proteins. This gene editing approach is still applied at the cellular model system, has not yet progressed to animal models in the context of ataxias or clinical trials. There are still numerous technological challenges that needs to be addressed to be able to move forward with this kind of therapeutic strategy. RNA therapies are being very popular, where with this therapeutic approach, we want to target mutant RNA transcripts. There's two main approaches, RNA interference and antisense oligonucleotides. So RNA interference, we want to silence the target gene either by degradation or by turning it off. This approach has been used in studies for many repeat expansion diseases, PLA, SCA3, SCA7, usually using patient-derived fibroblast. There's also studies for SCA1 in mice using a viral vector. And there's an ongoing trial using small interfering RNA for SCA2. One of the sites is the Neurological Institute here in Montreal. Antisense nucleotides will block, so it's a little synthetic molecules that will be linked to the gene, the gene of interest, and will block its expression. So we'll basically turn this gene off. There's preclinical studies that has yielded promising results for SCA1, 2, 3, 7, and 13. And there's a clinical trial that was initiated in 2022 for SCA3. So in summary, genetic and in terms of research for genetic investigation, I think this is a high priority for ataxia patient with familial history. We want to improve our ability to find the genetic causes. Some sequencing technology are not yet available in clinical setting, so we need to do proof of concept studies to demonstrate the efficiency of those technology and to prove that they can help improve the diagnosis. And combining different approaches, like I mentioned, a multi-omic approach will help to improve diagnosis yield and solve variant of uncertain significance. This is very important. Having a genetic diagnosis enables further treatment development. So we need to know what caused the ataxia. We need to understand what's happening to be able to develop effective treatments with the more precise technology. So this is a very important step that we're still working to improve in the research setting. Understanding disease mechanisms. So there's a lot of research on model systems using different model systems, and those are necessary to uncover disease mechanism. All those model systems have their own limitations. So usually using a combination of models can give a clearer and more precise picture. And these models, in addition to helping us understand the disease, are also useful to test drugs and for drug repurposing. There's a lot of effort for treatment development for rare diseases, including ataxias. The research is booming to develop and improve treatment strategies. And since oligonucleotides is considered one of the most promising approach, however, there's still some technical limitation of how it can be used on patients. And the model system and sequencing data will lead in the future to the identification of novel targets that could be applied to specific ataxia subtypes and hopefully to more broadly to different ataxias. So I would like to thank all the patients and the families that participated in those studies, my team, the clinicians that I work with on those projects. I will be happy to take on questions. I hope I was able to give you in 1 hour a picture of where the research is in terms of genetic model systems and treatment for ataxia with family history. So thank you very much.
Thank you so much, Dr. Tétreault. We will be focusing on ataxia questions related to genetic testing and unknown ataxia types with family history. If we don't get to your question, please feel free to email research@ataxia.org. And after this call, we'll be able to help you out. And with that, first we have a clarification question around some of the genetic technologies that you were talking about today. We have someone who shares that they were diagnosed with ataxia due to an SBG7 mutation back in 2019. He's wondering if he should be asking for long-read sequencing now that the technology has improved. Would that add anything to his diagnosis?
I don't think so. I think if he already has a clearer diagnosis with SPG7, I don't think long-read sequencing will bring more information. So SPG7 is usually a recessive disease, so you need two genetic variation. There is some literature that mentions that it can also be a dominant. So only one. However, this is not clear if it's true or not. So if for this person there was only one variant, maybe I would recommend it in this case to move to long-read sequencing to make sure that a second variant was not missed. But if it was a clear recessive SPG7, then I don't think long-read sequencing will bring more information.
Thanks for clarifying that. We have another community member who shares that her family is recently discovering that they have a history of SCOT27B, but not everyone in the family has the gene mutation. She's wondering if other family members who are experiencing ataxia symptoms but don't have the genetic mutations, if they could have a different type of ataxia, or how this might be explained with maybe some of the limitations in genetic technology.
Yeah, so that's a very interesting family. I would say it's not impossible that they have a different type of ataxia. Uh, would be unlikely, but not impossible. So I think it depends on which technology was used to diagnose all the family members. So maybe so a lot if it was by a lot are doing like repeat prime PCR or other technologies. So maybe that's a limitation of the technology. So maybe for them trying another non-clinical approach might be useful to really confirm if they do not have the mutation or if it's just that the repeat is a bit smaller. Because with repeat expansion, the size can vary within a family. We don't necessarily see the exact same size all the time. So that could be one reason. But we cannot exclude completely that they have a different type of ataxia, but I would say it wouldn't be my first hypothesis.
Thanks for clearing that up. We have an attendee who is interested in, well, a lot of what you talked about today is genetic testing. It involves uh, information both from the person being tested, but also their parents and their siblings. They're wondering if they engage with this type of research, could it come out that they are adopted? How do researchers, when they're engaging with this kind of research, handle these components of people who are adopted into families that have ataxia?
Yes, so for sure, yes, it could come out. Obviously, if we have genetic data on multiple family members, it's actually an information where we will see that. How we handle that? So honestly, in my experience, a lot of the time, if the the child is adopted and doesn't know, usually the parents won't agree to participate. Uh, because they usually know and they don't want to be in this situation. But if we have we're not supposed to disclose this information if we find it, because it can have a lot of consequences and issues that are will come from that. So usually it's not an information that we will disclose in a research setting.
And then speaking about research, we have an attendee who's asking if patients from the US can participate in the research that you're doing. And perhaps in addition to that, some of the technologies you talked about, how widespread are they in labs researching ataxia? So maybe a twofold question there.
So I'll start with the second part. So they're growing in popularity. For sure, in the US, there are many labs that are using these approaches. And we collaborate with labs everywhere in the world. Yes, there's a possibility of US patient participating in research in my lab or other labs in Canada in research projects. Uh, Often it's based on with a collaboration with a clinician or a researcher in the US because of ethics consent form that are can be more complicated. But we do have samples that are coming from all over the world, France, US, for our research projects. And I would say that usually in rare disease in general and in ataxia, research like is very collaborative. We work together. So if somebody wants to participate, just talk to your clinician, ask us to reach us, and we can find a way to include. And if there's any patient, RFC1 Canvas patient, we have a really nice project here in my lab with my collaborator. We're recruiting patient to study, so reach out to us or your clinician, ask your clinician to reach out to us.
And if you need help connecting with Dr. Tétreault, you feel free to email research@ataxia.org. We're happy to pass on any messages that we receive to Dr. Tétreault. And thank you for explaining that. One of the big questions we get all the time is how people can get involved with research. Uh, Moving on to our next question here. We have an attendee who's interested in how diversity considerations are taken into account with research. So when you're doing genetics research, what kind of variables are you taking into account when you're recruiting people for your studies and making these findings and conclusions?
Hmm. So diversity is a big problem in genetic research in general. A lot of the reference genome we're using to compare our samples are usually European descent. So when you don't fit in that box, it's more complicated. In our research, we try to be very inclusive and recruit all the patients, whatever the ethnicity, to include them in our project and to find ways to find controls um, that will match and give us a more accurate genetic picture. It's not always easy, but we're doing our best to do that. Being in Montreal and with the hospital here in Montreal, for sure, the community that is served here is mostly European descent. So those are most of the patients we include in our research are from European descent. We want to have the most information as possible, in clinical wise, and life habits, because all of this can, especially if we look at methylation, if we look at RNA, how you eat, if you exercise, all of this can impact what we see. So the more information we can have, the better. But it's a work in progress to include for diversity. There's a lot of group. There's a really big initiative, Canadian initiative to sequence 100,000 genome in Canada. And so there's 12 teams in Canada. My lab is part of this project. And the goal is to include indigenous samples. There's one project that has been funded is just Black community. There's an interest and an initiative to be able to include more diversity in genomic research in general, and that will help in the diagnosis of patients from those ethnicity in the future for sure.
Well, these are incredibly complex issues, so thank you for doing this research and giving us some answers to them. We have time for one last question. You've been in this field for quite some time. From the time you started maybe grad school to now, what has been the biggest change you've noticed in the type of research being done on ataxia? Has there been
Uh,
any major shifts that you've noticed?
my background is mostly genetics, so I will answer based on that. I think it's really the technology, how is that has involved. And I talked about the FGF14 families. I actually started studying those family in my master's degree, and we published them in 2023, and I was a professor at the university. So we've been working on those families for quite some time. So to be able to have the technology now to identify the genetic causes in those families. We tried everything. As soon as a new technology was coming out, we were trying it. We didn't find anything. So I think technology is really, in terms of diagnosis and understanding disease, is really what has changed the most. And now we have more tool, more power. But again, we need to be able to interpret the genetic variations that we see. So there's still a lot of work, but technology development has really impact ataxia research in general.
Well, thank you for teaching us a little bit more about this technology today and for your time and expertise, Dr. Tétreault. We are at time today, but thank you so much for joining us.
Thank you. Thank you for the invitation.
And to all of you who joined us for the past hour to learn a little bit more about unknown ataxia family history and the research being done in this area. I hope you learned something new about this area. And wherever you are in the world, good morning, good afternoon, good evening. Have a wonderful rest of your day. Take care, folks.